Good morning, everyone. It's my pleasure to introduce today's speakers for this joint GlycoNet ACS Carb Division webinar series. My name is Myles Poulin. I'm an assistant professor at the University of Maryland. It's my pleasure to introduce our first speaker, Theresa Reineke, who is a distinguished McKnight University professor in the Department of Chemistry at the University of Minnesota, and also holds joint appointments in the Department of Chemical Engineering / Materials Science. She received her PhD from the University of Michigan and then went on to carry out a NIH postdoc fellowship at Caltech before starting her own independent research career. She's done a lot of amazing work in the field of polymer chemistry and in developing new methods to make polymers, including glycopolymers, and working on ways of applying these for unique applications, such as in gene therapy, genome editing, and drug delivery. So I'm really happy to hear about her work today, where she'll talk about glycopolymers that promote delivery of functional nucleic acids.
All right. So today I'm going to tell you about my group's work in designing glycopolymers for delivery of functional nucleic acids. And, if you have any questions, I'm very happy to answer them. And what I can tell you is that my group really sits at the interface of polymer design, development, and sort of applications. And we look at utilizing powerful natural products such as carbohydrates for a number of applications in my lab, starting from sustainable polymers to try to replace petroleum-derived feedstocks with natural product monomers such as sugars, things from engineering interactions of polymers with small molecule drugs and trying to make them more bioavailable for oral drug delivery, as well as using them as delivery vehicles for really large payloads that have issues with stability and entry into cells, such as nucleic acids, proteins, monoclonal antibodies, etc. And today I'm going to really focus on this area of using our glycopolymer scaffolds for this important therapeutic modality. We're interested in things that range from fundamentals of synthesis, new methodologies, to looking at structure-activity relationships of these systems, all the way to applications, and so even tailoring things for tissue-specific delivery. And I'm going to get into that more towards the end of my talk. But just to give you a background in gene therapy, of course, many of us have become familiar with that, with the Nobel Prize being awarded this year in chemistry to the field of gene editing, which has been very exciting. And so, genetic diseases affect millions worldwide, and this is just a small sampling of over 10,000 genetic diseases that involve just even one monogenic defect in the human genome. There's many different strategies in which sort of gene therapy can be undertaken. And these are kind of three main, just to highlight how this would work. So for example, ex vivo would be where you would remove like a patient's cells. So for example, their stem cells or a CAR T cell or even skin cells. You would take that, grow it up in a tissue culture dish, deliver a functional nucleic acid into those cells, and then transplant those cells back into the body. And so that's called an ex vivo application. And of course, autologous stem cell transplants or CAR T cell therapies are kind of at the forefront of this therapeutic modality. Another is in vivo, where you would directly systemically inject the formulation into the tissue of interest and or even as a vaccine. So of course, DNA vaccines are at the forefront of COVID vaccines right now with Moderna. And lastly, one is where you would systemically inject, for example, into the vasculature, and it would circulate, and then a signal on that formulation would cause it to localize or be targeted to a tissue. And one example I'm going to talk today about is the liver, and it's a very important target for gene therapy. So right now, state of the art, and what I'll say is state of the art, but there's a lot of challenges, is to utilize engineered viruses for this purpose. And that's because viruses, obviously, over millions of years have evolved to infect human cells with their infective DNA and cause many types of diseases and illnesses. Of course, researchers can genetically engineer these viruses to remove the infective gene, put in a therapeutic gene, And, the surface and the capsids can also be further engineered to provide better internalization features and efficacy. However, if you take a look at all of the gene therapies that are clinically approved now by the FDA and on the market, you begin to see what the main issue, one of the main issues is that, it's the price. And you can see even Zolgensma has a price tag over $2 million per dose. This is per dose for one dose. To patients, which of course is extremely cost limiting, particularly with our difficult insurance market right now. So about 30% of this cost arises from virus development, 80% is, FDA review time. And one thing to note is that virus manufacturing facilities have queues almost as long as 2 years to even get enough virus to hold a clinical trial on these important drugs. So this is an important quote that, Bloomberg had, that gene therapy was hailed as a revolution and then came the bill. One other side effect is that things like AAV, the doses are huge. They've been shown to cause cancer in some clinical trials. So there's many things to overcome, including immunogenicity issues. So where my group fits into this is we're really looking to design synthetic materials, particularly using glycopolymers for delivery of these important payloads into patient cells. And today I'm going to tell you about work that we've been doing focused on degradability, targeting, and stability of these particular scaffolds. So the main question is, can we make gene therapy and all these important payloads affordable and available to the general public? And of course, if you think about, trying to translate this into third world countries, genetically engineered viruses are just impossible right now. So where the polymer and why a vehicle is needed is that, you take things like RNA, siRNA, messenger RNA, DNA oligos, plasmids, and even, CRISPR-Cas9, for example, plasmids that encode for CRISPRs, or even the Cas9 protein guide RNA, which are being also directly delivered as payloads for gene editing. All of these payloads are very sensitive to nuclease degradation, as well as do not get into the cell by themselves. So you really need to encapsulate them to promote stability and also promote interaction and internalization into cells. So we are focused at looking at cationic polymers that can promote an electrostatic interaction with these and encapsulate these payloads into nanoparticles. This nanoparticle, formulation is driven entropically by the release of counterions from these oppositely charged species. And then, we look to promote signals on the surface of these nanoparticles that can promote cell surface, package, payload interactions, internalization via active means through endocytic routes, such as caveoli and clasp mediated routes, internalization in the cells and release of these precious payloads. We've, over the years, looked at a variety of architectures, and a couple are, for example, alternating architectures where you have like this carbohydrate that's splitting up the charge in these oligoethyleneamines. Again, the charge here at physiological pH amines are protonated. They promote the cationic charge the polymer needs to interact with the negatively charged therapeutic payload, or block systems. And you can see we have either carbohydrates polymerized along the backbone as part of the backbone, or carbohydrates pendant off the backbone that are creating this sort of block copolymer structure. So a block of carbohydrate and then a block of charge. And I do want to note that we varied charge in pKa. And so there's like all sorts of variables that we've looked at with these, as well as different carbohydrates, from linear monosaccharides to disaccharides to things that hang off the backbone, mono or di, or even things like N-acetylgalactosamine that can promote cell specificity and interactions with various epitopes. We've also looked at many different architectures, so things that are like alternating, block, things that are star branch, that micelize. I don't have time to talk about all of these, but I'm really going to show you some work and talk about our work that's ongoing and like kind of creating and comparing linear statistical and block architectures. So where this is important and why this is important is that, alternating structures can easily electrostatically interact with these payloads. and compact these payloads into these sort of nanostructures that have the charge very close to the surface. Block copolymers, you take that cationic block, it can encapsulate the payload into the center. And so you get these sort of core shell architectures where the shell consists of a polymer brush of some hydrophilic modalities, such as a polymerized carbohydrate. And when you formulate these nanoparticles in water, you can see this as a transmission electron microscopy image of how they look. They're small, 100 nanometers or below, similar in size, shape, morphology to viruses. Yet when you put them into biological media that are buffered, contains a lot of salts, physiological pH of 7.4 with 150 millimolar of sodium chloride, you can see that these condensed structures actually aggregate very rapidly, almost into micron-sized particles, whereas these core shell structures can remain colloidally stable, which is very important if you're going to inject something like this into the bloodstream that needs to circulate and potentially hone into a tissue target. So embarking upon this, like early on in my independent career, we really had the hypothesis. So polyethyleneamine is, a polymer of ethyleneamine, and it was been well known for many years to promote packaging of these payloads and cellular internalization and delivery. However, it's quite toxic. So we had the hypothesis that, maybe we could copolymerize a sugar that's, benign, hydrophilic, with something that has a PEI-like block to create, polymers that have this charge density broken up, yet could still encapsulate and promote delivery of these systems. And so this is one variant we named glycofect, and we've kind of arbitrarily named these polyglycoamidoamines, or PGAA scaffolds, where we have this sort of hydroxylated monomer that is copolymerized with various lengths of other comonomers of secondary amines. There's many different architectures where we varied stereochemistry of the sugar, number of hydroxyls, various lengths between 1 and 6. We have linear and branch variants that we've created almost about 50 of these different architectures to really explore all sorts of different interactions. But in a nutshell, all of these polymers encapsulate these payloads, they deliver them into cells, and often, we use DNA that once it gets into the cell, it encodes for a reporter gene, such as green fluorescent proteins. So if the polymers deliver the DNA into the cell, it actually expresses GFP, and we can do a lot of, fluorescence intensity and cell counting just to look at, as an assessment of whether or not the polymers work or not. And then we do a lot of work in toxicity. One particular structure that contained galactose ended up being the best and can deliver DNA to almost 100% of many different cell types. And we've looked at, hundreds of cell types, and it worked quite well. And interestingly, these polymers, even though they are nylon-like derivatives or polyamides, we discovered that they actually do hydrolyze under mild conditions over a relatively short period of time, enabling a hydrolysis and degradation mechanism after they deliver their payload, and a means to actually release their payload once they get into the cell. We've also done a lot of work looking at how these interact with cell surface proteins, how the cell actually internalizes via caveolate-clathrin routes by doing a lot of immunohistochemistry and trafficking work to basically show that these nanoparticles do interact with, are internalized, and are trafficked under very similar mechanisms as viruses within the cell. And even as far as looking and showing co-localization with intracellular organelles like the trans-Golgi, and even retrograde transport to things like ER, like the endoplasmic reticulum, again, similar to active transport mechanisms in which viruses show. And this is just one kind of snippet of data showing that, comparing the glycopolymer with the PEI that has no carbohydrate, that in fact, we can dye the DNA, we can do a lot of immunohistochemistry where we're using antibodies to tag. This is endoplasmic reticulum, doing colocalization and really looking quantitatively at colocalization, showing that, the presence of carbohydrates, in fact, does help with mediating active transport mechanisms more similar to viruses than without carbohydrates. So we've taken, done a lot of work. And again, I'm summarizing sort of some of our earlier work here where, again, they get into cells. We've shown like kinetic transport data where, like the the DNA and the polymer enter the cell, they're together in a package. And then after a while, you can see this M2 decreases. And in fact, you can see the DNA here is released, it's in the nucleus, and the polymer appears to actually be ripped or released from the DNA. So just showing kind of kinetics of this process and release once they get into the cell. That G4 variant early on was actually commercialized by a small company called Teculon to be a mild non-toxic delivery agent to very sensitive primary cell types. And in fact, we've also done a number of animal studies to show that even using this delivery vehicle through direct injection to tissue types like the mouse heart, we can actually deliver nucleic acids that block COX-2 and even have a protective effect for heart attack victims, or even decreasing damage to heart for things like hypoxic conditions that heart disease or heart attack victims can experience. So with that, again, kind of that's some work that we've looked more at in vitro ex vivo work. I do want to highlight some work where we've used glycopolymers for in vivo applications. And I do want to just note as kind of a snippet that we've looked at trehalose, which is a disaccharide that's well known for its high hydration sphere, as a replacement for things like polyethylene glycol to promote highly hydrated systems that can, encapsulate the payloads, be lyophilized to dryness and storage, be rehydrated, and still retain biological efficacy of those really sensitive payloads such as proteins, nucleic acids, and even show efficacy in vivo for delivery. And delivery of these important payloads to liver. And so the liver and liver hepatocytes, and hopefully I'm seeing my internet connection. Hopefully I'm still, yeah, you can see me. I'm getting some messages. My stability is not well, so please feel free to pipe in if I'm cutting in and out. But liver hepatocytes are an important gene therapy target because the liver is essentially, an organ that excretes our serum proteins. So it's well known to excrete many different proteins. But with that inherent mechanism of this important organ, we can also sort of, utilize this beautiful inherent excretion and by doing liver gene therapy such that, for example, for people that need sensitive protein therapies, so for example, in hemophilia, Hurler syndrome, various other lysosomal storage diseases where people need a constant source of protein in their blood system, doing liver gene therapy instead of constantly taking or getting injected with these proteins could be a long-term solution to many of these genetic diseases. If you could hijack part of that serum, even just a small percentage, such as 0.2% of liver hepatocytes, they could serve as an in vivo factory just to create and excrete these important proteins that people are lacking in these genetic diseases. So the important thing is that liver hepatocytes have what's called the asialoglycoprotein receptor on their surface. And the question we sought the answer was, if we sort of display a signal that could promote selectivity of delivery to these important cells, hepatocytes, can we promote internalization in vivo of important genes to give a potential mechanism for doing targeted liver gene therapy to hepatocytes. And so, acylofetuin, GalNAc, galactose, lactose are many sugars that are well known to target the ASGPR. And today I'm going to show you that polymerized GalNAc has an even much higher affinity to do this, and important work that we've done that's shown we can actually promote specificity. So I'm not going to go into detail with this, but what I do want to highlight is that we have made a methacrylamido GalNAc monomer. And in fact, this shows a several step process, but we have decreased this into just one step recently in my lab to go from GalNAc to actually work that we've selectively functionalized this anomeric hydroxy with this monomer. We then take that monomer, whether it's methacrylic or methacrylamide or acrylamide, we can do a polymerization strategy called reversible addition fragmentation chain transfer polymerization. So we can actually dial in almost exactly the amount on average of these monomers that are hanging off the backbone in a pendant manner. And our publication a few years back explored, a couple of different strategies to look at lengths of the GalNAc block that are shorter, about 20, all the way to things that are about 80. And then hanging off further chain extending with different block lengths of this charged amine structure. And so all follow-up work that has been recently submitted as we've looked at a whole library of different polymer and monomer lengths of the GalNAc block with this different cationic block. In this work, we felt it important to make some controls that contain just glucose, as well as a polyethylene glycol. So this would be the targeting block with the GalNAc, and then the non-targeting controls of a glucose and polyethylene glycol. So what we've shown is we've taken those blocks, and this is dynamic light scattering data to look at hydrodynamic diameter of the complex into the polyplexes, and whether or not these complexes actually are sort of discrete entities, are non-aggregating, or do they aggregate into big structures? And we want to avoid the aggregated structures. And so what you can see is throughout most of these GalNAc polymers, we actually do get pretty stable. We can form the polyplexes. They're pretty stable in the cell culture media. We've done light scattering in to show that the colloidal stability is still promoted by displaying that carbohydrate on the surface of our core shell structures. What you can see is that, in fact, the ethylene glycol derivative actually has slight lower colloidal stability over time with that, but still pretty reasonable for our PEG control. Cytotoxicity, so just exposing cells to these nanoformulations, we show that the N/P ratio I want to note is just a formulation ratio. It's the number of nitrogens from the cationic block to phosphates on the payload or plasmid DNA that we're using. And what you can see is we increase that ratio. So for example, 7.5 means 7.5 times more nitrogens or polymers to the DNA. So as we increase that formulation ratio, you can see we do get more cytotoxic effects, just denoting that excess polymer in the formulation can actually have a detrimental effect to the cell health. With respect to sort of interaction of that acylglycoprotein receptor, what I do want to note is that we've looked at, for example, glucose, which is the yellow bar, galactose, which is this turquoise, GalNAc monomer, acylofetuin, which is a protein that targets the ASGPR, as well as our polymerized GalNAc. And you can see that the IC50 value significantly decreases, and we're getting much tighter binding by polyvalently displaying that GalNAc on that carbohydrate block of these formulations to show that, the receptor binding is much, stronger. With respect to specificity, what we did is we, again, formed the nanoformulations. We then exposed both HepG2, which is a hepatocyte cell line that we cultured in our lab, and then a HeLa cell line that does not contain the ASGPR receptor. So both with a cell line with and a cell line without. And what I want to note is that without GalNAc block, so our controls, which are this PEI, jet PEI, and the glucose block polymer, you can see that in fact, the HeLa cells actually have more DNA and higher gene expression, as well as that polyethylene glycol block. So you can see that there's not really any difference. There's a slightly statistically higher difference in the PEI block for the HeLa cells, the cancer cells. However, with the block polymers that displayed the multivalent display of the GalNAc, you can see that there's statistically higher internalization and gene delivery with the hepatocyte cells, showing that, you know, we have promise that these formulations do have some specificity into these liver hepatocytes. Furthermore, we went on to do tail vein injections to follow kinetics of liver localization. And so what we did is we took those block polymers, we tagged them with a near IR dye, so size 7, and our polyplex formulations then were fluorescent, and we could track localization into the mouse liver. And so you can see within 30 minutes, these formulations do get to the livers of the mice. And then you can actually see starting to sort of be eliminated through the kidneys of these mice over a period of hours and in fact a day. So it does appear that these get into the liver. We also further sort of excise the organs, and you can in fact see that the liver, as well as the kidneys, do internalize these formulations. And we do get, you know, in the liver, when we quantitate the harvested tissues, we are in fact getting a much higher statistically significant difference of these formulations containing GalNAc compared to other controls like the glucose formulation, as well as comparing it to the other organs in the mice. So with that, that shows you kind of a small snippet of the work we're doing in our lab with respect to glycopolymers that showed that, you know, we can create these polymers for these very important therapeutic and up and coming payloads for many diseases that promote degradation, tissue targeting, as well as the trailose vehicles promote pretty exceptional stability of these payloads for potential storage. And with that, I'd like to thank you all for your attention and thank a number of people in my group. This is, of course, my current group, but, you know, there's been many people throughout the last, 18 years that have contributed to this work in my group. And thank you all for your attention. I look forward to hearing questions.